Hi, this is Yoyo. This video is my final presentation for my Biology 110 course at Duke Kunshan University. However, I do hope this to be interesting for everyone to watch. So I tried my best to add some humor in this, and I hope you enjoy this. So let's get started. Today I'm going to mainly talk about diabetes, insulins, and cell transformation techniques that are going to produce insulin to treat diabetes. So diabetes, as the definition from the WHO, is a chronic metabolic disease that suffers around 422 million people worldwide, and the number is increasing. The features of this disease is the increase of blood glucose, or we can also say blood sugar. This increase will cause damage to vessels, neurons, kidneys, and many other tissues and organs, and in some cases it can be fatal. The main two types of diabetes are type 1 and type 2. Type 1 are usually discovered among youth, while type 2 are more common in a larger population. Diabetes are usually caused by either an inability to produce enough insulin or the body's resistance to insulin. In many cases, the treating of diabetes include proper exercise and nutrition intake under the instruction of a dietitian, and proper medicine use under the instructions of doctors. Injecting insulin is also a commonly used method for many patients to control their blood sugar effectively in their daily lives. So today we're going to talk about how are we going to produce sufficient insulin to treat diabetes. As we already know, it's unethical to extract insulin from other living human beings, and it's quite not easy and rather expensive to produce enough insulin for medical use through chemical ways. So how about doing this in biological ways? For example, can we use bacteria to produce human insulin? Then we'll be faced with two problems. First, which bacteria are we going to use to produce safe and usable insulin? And second, how are we going to get the bacteria producing human insulin? As our knowledge about genes develops, scientists have already come up with methods to extract genes by cutting them with enzymes from the chromatins at the nucleus of the cell. This cutting will result in a gene fraction with two sticky ends, which enables it to be incorporated into other DNA sequences and therefore might be replicated and expressed successfully in other organs, such as a bacteria. All this can be done in the cell transformation that I'll introduce to you in a second. The most commonly used material for cell transformation is E. coli, a kind of bacteria that can be found in many places, including humans' large intestine. Some of them can cause disease and some of them cannot. The reason we use E. coli in our cell transformation experiment is that E. coli contains two different types of DNAs. One is what we often see in many cells, and the other is what we call plasmid. Plasmid is a ring-shaped small DNA that is much smaller than the other DNAs we see regularly. And if we zoom up a plasmid, we're going to see several basic structures. One is called origin, or shortened as ORI, which is the starting point of the replication of the entire plasmid. And the other is MCS, the abbreviation for multiple cloning site. This area can be recognized and cut by many different enzymes, forming an opened ring plasmid welcoming the joining of other DNA fragments, such as the gene we need to produce insulin. One other structure that can be found in many experimental use plasmid is one antibiotic resistance gene. As the name shows, this gene will help the bacteria containing this plasmid survive under the condition with antibiotics. But you may ask, why is it there? Please remember this question because we're going to talk about the function of this antibiotic resistance gene in several minutes. But now let's focus on solving another problem. Do you think there will be any problem in the combination between the open ring plasmid with the gene we need? Well, actually, there are two possible problems. First, The gene is not incorporated into the plasmid because the plasmid is cut by the enzymes that we talked before, and therefore it also has two sticky ends, which makes the plasmid very likely to form a ring by itself again. Another problem is that the gene might be incorporated into the plasmid reversely, which means although the gene has been incorporated into the plasmid, it cannot be expressed properly because it is reversed. I know this is pretty puzzling, but let's do an in-person illustration to get the idea clearer. So, this is me imitating a plasmid, and as you can see, cutting by the enzyme, the plasmid opens up and having the chance to accept new genes. However, there might be cases that the plasmid goes back and forms a ring by itself again. And another problematic case is that the plasmid forms a ring with the gene, but the gene has been incorporated reversely, which means during the process of transcription and translation, the information on the gene sequence will be read reversely, and therefore the protein produced in this procedure will likely be malfunctional. So now we need to solve this problem. How can we determine which plasmids incorporated with the gene correctly and which did not? Well, scientists have come up with a method called PCR, the abbreviation of polymerase chain reaction. It is a method to mass replicate short DNA fragments in relatively short time. And since it largely increases the amount of the DNA fragment to be tested, we can make easier observation of the DNAs. For example, one commonly used testing method of the DNA fragments is to use electricity and a special gel, which can make the DNA fragments run in the gel, and the longer DNA will stop earlier, and the shorter DNA will stop later, and therefore we can determine whether the right DNA fragment has been incorporated by its characteristic length. Now we get the plasmid containing the gene we need, which means the gene we need now can stably exist in new cells and can be accurately replicated using the new cell's polymerases. The new question is, which cell are we going to use as the acceptor of this plasmid? Well, as you may already guessed, E. coli again. After treating E. coli with calcium ion and heat shock, it can become competent, which means it's able to take up the plasmids in its surrounding environment. And after putting the plasmid that we just get into the liquid containing these competent E. coli cell, we're going to get E. coli transformed by the plasmid, or what we called transformants. However, there's yet a problem. As shown by me, although the plasmids can get into the cells and transform the cells will not look different. Just like the library building I was in. Although I am acting as a plasmid in the library building will not look different, and it cannot be differentiated from other buildings. Similarly, transformants can also not be differentiated from the untransformed E. coli cells. So how are we going to determine which E. coli cells contain the transformed plasmids and which do not? Well, do you still remember the question about the antibiotic resistance gene? This is exactly what it is for. If we incubate both transformants and untransformed E. coli cells in an environment containing antibiotics, such as ampicillins, then the transformants will survive and form cell colonies, while the untransformed E. coli cells will die and no colony will be formed. However, have you ever been to a biology lab before? You may realize that during the process of incubation or even storage, not only your place will be incubated or storaged, but also many other people's plates containing many different kinds of cells colonies, and it can be difficult for you to differentiate one plate from the other. So, although you may resolve the problem by labeling your plate, you still want to make sure that you can recognize your plate at the first sight and therefore prevent any confusion. Since we already used the Duke Kunshan University's library to illustrate the idea of a cell, Let's just use the entire Duke Kunshan University's campus as a cell colony. As you may see here, if we consider this is what a regular cell colony looks like, if we don't make any adjustment, a transformant cell colony will also look like this, and there might be confusion. So, are there any way to make the cell colonies of the transformants to look different? Well, scientists have come up with an idea of making a special kind of plasmid for transformation. The one that we used in our experiment is PUC19. It contains not only the antibiotic resistance gene, their origin, the MCS, but also a special gene called the LAC-C gene. It encodes the beta-galactosidase enzyme. This enzyme can catalyze the hydrolysis of the substrate X-gal and therefore create an insoluble blue pigment and leads to the blue cell colonies of the transformants that can help the differentiation between the transformant cell colonies and untransformed cell colonies. Finally, we get the transformants we want, and these transformants can then be used for mass production of the cell products that we need, such as the insulin. Of course, apart from producing insulin, the transformants can also be used to store and contain certain genes for study, or be used to study the expression of other traits of the gene. So the most valuable person here is E. coli. Thank you so much for your attention, and my genuine acknowledgement to Professor Lee at BIOL 110 at Duke Kunshan University, as well as my dear school Duke Kunshan University as acting as cell colonies in my video. And this is my reference list.